Good morning, everyone. Thank you, first of all, to the organizers for this wonderful conference. It brings me back in contact with one of my collaborators I met almost 10 years ago, and I'm going to talk about that work, which is my only paper that have something to do with evolutionary game theory. So the title of this talk is part of the title of that paper, The Evolution of Cooperations in a Two-Level Moran Model of the Snowdrift Game. So I'm going to quickly talk about some background, maybe within group dynamics of the Moran model. So this is the paper I'm going to talk about. So my talk today will focus on basically summarizing this paper, 2018, you can see, with Brian McLoone et al. And Brian was leading this project. At that time, we were both postdocs in different labs. And so basically, I will quickly summarize what we did in this paper. And we'll focus on the stochastic model, the two-level Moran model. I view this talk might be as a sequel of Dan Cooney's talk and also Dan Chan's poster on two-level selection models. And this work come from discussion with Lawrence Lovey, which is a PI in the Wisconsin Institute for Discovery. And at that time, we're talking about modeling microbiome interactions and evolutions within lung tuberculosis. So I guess this discussion is more about biology inspiring math and less about math inspiring biology. But I'll try to touch upon the latter as well. So basically, we're talking about, in tuberculosis infections within a human host, a lung, there are granulomas, and then within the granulomas, the bacteria might produce some chemical and biofilms to catch ions, which they need. And the thing is that we use Snowdrift Game because the idea is that the cooperations within the granuloma is costly for individual bacteria, but it's not completely sacrificial. And so Snowdrift Game seems to be a sort of a makes sense first approximation within a granuloma. And we're interested in understanding selection, whether it occur both within the granuloma and also between granulomas. And this is very high level discussion. We also talk with other microbiologists in the Wisconsin Institute for Discovery, which would probably lead to a sequel paper, which never happened. So that's why I say it's more about biology inspiring math. So just a quick recall of this Snowdrift game. It is, imagine two drivers trapped by a snowdrift, and then, if they both cooperate, or if any one of them cooperate, they go home. If none of them cooperate, then no one go home. So the two-player payoff matrix is down there below, right? It's B minus C over 2, B minus C, B and 0. Well, an embarrassing notation we used at that time is we did not use our RSTP. We use alpha, beta, delta, gamma. So you can see. So just a quick recall of the within group two-player Snowdrift game, a fitness of a cooperator inside a group is given by this, right? And the fitness of a defector is this, just B times X, where B is the benefit and C is the cost parameters. And the average individual fitness is given by this W bar, and this W bar will appear in the group level Moran process later. We know there's a replicator dynamic, so if the group is well mixed and infinitely large, right, we often use this ODE to describe the frequency of cooperators within a group. And you might notice that there is a fraction, this W bar here, and sometimes it might not be there. In Dan's work, I don't think it's there. It just depends on how we scale, how we scale the Moran model underneath. But nevertheless, whether we have this W bar, they have the same equilibrium. And for Snowdrift Game, there's an internal stable equilibrium, this X star. So how do we understand it in finite group size capital N? So capital N is a finite group size. Well, sometimes I think we use little n, but let's say it's capital N here. And the within group is a Moran process. In the paper with Brian, we use discrete time Moran process, but we can also use continuous time Moran process. And so, the number of cooperators, C and K, we increase by 1 with this probability in the next step, or decrease by 1 with this step. And the precise connection with the replicator dynamics, xt, is given by this, right? So for any fixed time compact time interval, we have uniform convergence almost surely. And so we can also consider continuous time process. I think in Dan's work or in some other work also, people consider where individual with strategy Y, where the cooperator or defector, will reproduce at a rate, exponential clock rate, right? 1 plus this Wi times pi y. Pi y is the individual fitness, and Wi is a parameter capturing the selection coefficients, right? And when we say weak selection, this Wi might be of order 1 over capital N. We also look at, so the ODE does not give us like fixation probability or any stochastic effect. So if you want to look at stochastic effect, we need to either go back to the Moran model itself or do some kind of approximation. Diffusion approximation. One standard way was done by Arna before, and it is basically have a correction term that is of variance order 1 over n. So one can look at a stochastic differential equation that look like this to approximate the Moran model. And the precise connection is similar to the one I showed before. For any compact interval, the Moran model, the fraction of cooperators will be close to this quantity x and t. And note that there's no weak selection required here. All right. So if there's weak selection, then one can obtain another stochastic differential equation, another approximation. So I think, so what we did is, now we connect to stochastic. So the reason to connect to diffusion approximation is that we can use stochastic calculus. So for this SDE in general, we can look at the first time, tau, where the frequency hits 0, 1. And then ask what's the probability it hits 1 before 0. And we call this function h of x, where x is the initial frequency, right? So h of x is the probability of cooperation go to fixation starting at x. And this h of x turned out to solve basically it's a Green's function. It solves a harmonic equation with boundary condition 0 and 1, strictly increasing. And so it can be solved to be this one, right? And now, so what we did in this in the paper with Brian is that, so this is a graph of this h of x, right? As we know, it's to be increasing, and we plot it for the parameter b equals 10, c equals 3, right? And we also plot it for different group size, 10 or 100. We see that when n is 100, big enough, basically it goes to 1, basically. It's basically almost 1 everywhere, meaning that no matter what the initial condition. And what we found for the Snowdrift game is that there is a threshold cost to benefit ratio we call R star, below which the fixation probability is basically 1 everywhere, right? Except at the boundary. But in theory, it's basically 1 when n is large enough. And when the C over B cost to benefit ratio is bigger than R star, then it's basically 0. So there's this kind of dichotomy of fixation probability. This is kind of interesting because if you look at two-level model, there are groups within group dynamics and between group. And basically it says that if the within group dynamic is fast relative to between group dynamics, then after some time, all the within group are fixed or the fixates depend on their R, right? No matter what the initial condition is. So And then, after that, every within group got into fixation, and then between group occur, right? So we can look at, we can think in this way. If we have, in the extreme case where between group is so slow compared to within group. We also actually did some work not in this, put in this paper, but if you look at stack hunt game, there's also similar dichotomy, not 0, 1, but it will be, in stack hunt game, there's also internal, under mild condition, internal equilibrium, which is unstable. And then the h of x will look like this, 0 and then 1, right? I forgot the boundary point, but it's like that. So in the two-level Moran model picture, now we have to care about the initial fraction of cooperator, not just the game matrix itself, but we still have this kind of dichotomy. So after some time, we know which group will fixate to cooperation, which group will go to all defectors, and then so it gives us something, right? So now let's go to the two-level model. The two-level model, this picture, I think we see something, Dan talk about this picture as well. So within group selection, so individual reproduce just like before using individual fitness, but between group will depend on the average fitness W bar, right? So in the paper with Brian, we look at, we consider discrete time Moran model, but we can also consider continuous time Moran model, using this group level average fitness. So this is the model. We every discrete time, we specify whether group J will reproduce or not. And then if it reproduces, it replace one of the other group uniform at random. So it is the capital B is the birth date update Dan mentioned. So we did simulations in this two-level model and look at the average frequency of cooperation in the entire so-called meta population, right? Every all the groups and all the individuals. For different R value, R is C over B, and different group size, and from 10, 100 to 1,000. So basically, this is a simulation study, and we found that for small group size, the average level of cooperation seems to be enhanced relative to bigger group size. And for bigger group size, the bottom row here, we see some kind of stabilization here. Well, after this work, right, because this was 2018, and then I think Dan's maybe 2019 or something, right? Obtain a PDE for this this kind of two-level Moran model, and from that we get new insight. We know that the group replication rate has to be fast enough to see sort of enhanced level. And so in the simulation here, because our group level replacement is not fast enough, so the average fitness is still the same as this dotted line. So there's a dotted line here, which might be hard to see, but which is just the average equilibrium given by the replica equation. So it's sort of the null hypothesis. So let me talk a bit about the insight from Dan's work using the PDE limit. So let's say we consider the Moran model now with let's say continuous time birth that update, where individual of strategy Y reproduce at a rate 1 plus Wi times pi y, where Wi is the number controlling selection within group selection coefficient. And then between group, it reproduce at, also 1 plus Wg times G of x. So G of x is, in our case, is the W bar. All right. And so then obtain this PDE, right? by looking at the fraction of groups with X% cooperators. And this FTX will describe that limiting frequency across all groups. And then with Yoshiro thing, right, then obtain that there's a critical lambda star, where this lambda is the relative group replacement rate and within group replacement rate, right? So this so lambda is this one. And if lambda is strictly bigger than lambda star, then for Snowdrift Game, there's expected enhancement. But when lambda is smaller than lambda star, then it does not show any enhancement relative to the within group dynamics. So in other words, our simulation in the previous slides is just that we might need to do more simulation when the between group is fast enough to see enhancement. So I guess this is, I don't know, mathematics inspired simulation study, not biology. Our paper with Brian also did some other simulation study where we allow, different game matrix with among groups. So we look at different BJ and CJ, in different groups and see more variation. But in general, we see some enhancement of cooperators frequencies when the group size is smaller, the within group size is smaller. So I've told you what we know so far is for this two-level Moran model. And what we don't know is that the PD does not tell us the fixation probability, any stochastic effect like a fixation time, quasi-stationary behavior, which is what we see if we conditioned on non-fixation. We also, if you pick a sample, gene genealogy of a sample of that population process, I don't think we know. We don't know the site frequency spectra for that sample genealogy and the multi-level selection. Although technically it is some kind of structure population process, right? It's just a special kind of structure. So Max has a very nice paper on structure coalescent. I encourage you to take a look. We also don't know effect of small n and m, right? Which is, hard. I don't think any analytically thing have been said, right? So let me spend, I think I have 5 more minutes or so. Okay, great, thanks. So what can we do, right, in the future is like, maybe for the forward-time two-level Moran process, one can deduce some robust scaling limits, right? PDE limits deduced by Dan is great, right? It can give analytical insight. And there are other scaling limit one can do for the two-level process, which I will spend maybe 1 or 2 slides to talk about it. So there are 2 papers very great, by Shishilo and Law and Mattingly, and also later by Valerit, about different scaling limits of a very simple two-level Moran model. And we can then analyze those limits, right? So the reason to look at scaling limit is that they are sort of believed to be robust in the sense that if you change the microscopic model a little bit, right, you still get the same limits, just like central limit theorem is. There's some kind of universality that is sort of believed to be there, that we don't need to know the nuances of the biology. We can still study that scaling limit. Another reason is that usually scaling limits have more mathematical property, which is they have more symmetry, they have more scaling properties, so they are more elegant mathematically. So I want to mention this paper by Donald Dawson on multi-level selections. He looked at more general models, maybe even 3 levels, and with migration, with selection and mutations. And he constructed this set value dual process of those multi-level, multi-scale, multi-type selection processes. So, and there's also another approach to modeling, which is sort of the retrospective approach, where we look at sample in a recent or current time, and then ask how are they related backward in time in the past. And this might be a more natural approach if you want to compare with genomic data, right? Population genetic people often do this. So this direction might be one way to look at how math inspire biology, right? We study how those random trees look like, right? I don't know if people study those. But for the two-level selection, two-level Moran model, one can imagine a coalescence process is not that, how to say, one can sort of guess what it might be, right? Like within group, it depends on the relative rate of between group replication and within group replication. But think about the extreme case where, every sample now, if you pick 5 sample in a large population, some are in the same group, some are in different group, then your sample will also have a label that tells you which group it is in, right? Or are they in the same group? So there's extra label for those samples. And then those label can also coalesce due to group level replacement event, right? So one can sort of write down what is the rule it should be. Now, would that be a robust scaling limit like a Kingman coalescent? It could be, right? Like within group dynamics, when the number of, the capital N goes to infinity, if you scale time to be 1 unit of time is n generations or n squared steps in the Moran model, then you expect Kingman coalescence to arise, right? And so, the label can also coalesce according to Kingman if you scale time suitably. So it's a mix. It can be a mixture of those. So, and you get a random tree, and then now you can study branching of that random tree, and then study SFS, and then compare with data. Yeah. Anyway, so let me spend the last 2 slides on talking a little bit about what kind of robust scaling limit one can obtain using the simple model by Shishilo and Jonathan Mattingly. So let's just quickly recall what is the model of Law and Mattingly. Right. So it's just it's a 2-type continuous time Moran model with 5 parameters, and little n is our capital N, is the within group size. M is the number of groups, and then there are selection parameter S and R, and there's a time scale, capital lambda. So within group is every type C replicate rate 1, every type D replicate at rate 1 plus S. S is a selective advantage. Between group is every group. Oh, so after replication, you just choose one of the individual within the group to replace. And then between group is the group J will replicate at a rate that is 1 plus R times Xj, where Xj is a fraction of type C. So it is just this simple model, right? And then what Shishilo and Mattingly show is that they, under different scaling, they obtain different scaling limits, including a PD, right? So let's talk about just the PD. So what they actually look at is this empirical measure. Basically, you can plot it as like a histogram. They look at a fraction of groups with Xj many fraction of type C. So they look at this. So this is, so they put a direct delta at that location, and then normalize it by the number of groups, right? So so this is a measure, and it's a Markov process, measure valued Markov process. And they show that when n and m converges to go to infinity, then this random measure converges to a deterministic measure that has a density that solves this PD. So that's what they show. So that's kind of, so I think, right, yeah, so this is the PD. So what about other limit, right? So there's several directions. One is, one can look at, one can subtract the Moran model by this PD and then scale it and then get a central limit theorem. And that would be a Gaussian process. That's one way. Another way is to look at weak selection regime where, they can get a so-called measure value diffusion limit. In that weak selection regime, we look at the selection coefficient to be of order 1 over little n. So let's call it sigma over little n. And R is the group level selection, which is also of order 1 over m. And then they also let n over m goes to a parameter called theta. It's like the ratio, right? And that theta will appear in the macroscopic limit, in the stochastic PD limit. And so what Jonathan Mattingly and Loss show, they show that this random measure, again, converts to now another random measure, mu t, which is a type of Fleming-Veal process characterized by martingale problem. They did not write it in a stochastic PDE, but I think one can check that this stochastic PDE is exactly the same as the Fleming-Veal process they wrote down. I write it in a stochastic PDE form because it might be more direct, right? We don't need to write down the quadratic variation of the martingale, but it looks like this. So under this weak selection limit, but we don't forget we need to speed up time by n. So the time is also sped up. So this is kind of like a Wright-Fisher, diffusion kind of scaling limit. We have to speed up time, and then we get this stochastic PD limit. And then the good thing is that one have a duality for this kind of processes, and then one can use duality to analyze maybe extinction probability or other behavior. I know this I have not studied this yet. This because I prepared for this talk to write this down. But I want to show you that some analysis can be done for simpler stochastic PDE. And so it is a spatial version of the Wright-Fisher diffusion, right? So this is a stochastic PDE called Fisher KPP stochastic PDE, which is like a spatial version of the Wright-Fisher diffusion where the individuals are migrating very fast. It's like a limit of the stepping stone model. And then there's a space-time white noise W dot involved to control the variance. And then one can say, one can prove various things of it. For example, one can compute fixation probability to be, explicit and, very simple. It is remarkable. Usually for spatial system, we don't have explicit fixation probability. And the proof is rely on the duality. So the duality in probability is that this stochastic process UT will be due to another particle systems. And then one can study, use the stationary behavior of the particle system to get extinction probability of this system, of this process U. And there's also unique quasi-stationary distribution, and we can obtain asymptotic for the fixation rate. Fixation rate here means that probability of fixation time bigger than t, when t goes infinity, will be like either the minus that thing, that. So that's what we obtain. We can sort of get some idea of what the fixation time should be asymptotically. Yeah, so I think I'll stop here, and thank you.
One question before transitioning. Yeah, okay. So in the initial part, you mentioned kind of that you're not using weak selection, but you're still working on the Snowdrift game and you're interested in fixation probability. But then somehow kind of you might get issues with fixation time, right? Because usually it takes a long time. Then is this a problem that disappears as soon as I start with PDEs, or?
That's a good point. Yeah. So we did not look at fixation time, but you're right. Because there's a stable internal equilibrium, fixation time should be exponential in n, right?
Yeah, but you could still do kind of, not really weak selection, but something intermediate, right? You don't have to wait forever for fixation. So it can still happen, and even if you don't send W to your intensive selection to 0. And we can talk about this later.
So yeah, I think let's talk about it later. So yeah, we don't look at fixation time, but you're right. How much we can sort of utilize within group, fixation, probability or the knowledge there to infer, what happened in the 2-level model. It depends on the fixation time within group, and we did not look at it. But in one-dimensional system, one can get some asymptotic, but one can say something about the fixation time. Yeah, right.